In this video, we will discuss about Dasatinib can inhibit the stem cell generations that are usually induced by TGF-beta, and also the TGF-beta induced migrations or cell motility. So, in order to check this, whether the Dasatinib inhibits the stem cell generations, so what they did is they expose the PANC1 cells for like 14 days under TGF-beta, and it will lead to the upregulations of the various stem cell markers such as beta-activin, BMP2, CD90, CD105, OCT1, and the E-catherine markers goes down such as NCAM1, the TGF-beta induced conditions, and surprisingly they also see a decrease in the amount of ABCG2. So this data shows that there is upregulations of the stem cells markers or stem cell generations is increased when the cells are exposed, when the PANC1 cells are overexposed to the TGF-beta, and in this case the exposure is done for 14 days, and after that or parallelly they also did the experiment in the conditions where they not only use TGF-beta but also use Dasatinib, and in that case they are able to see a suppressions of the stem cells markers. For example, in this case of this beta-activin, there is a great suppression. There is a significant suppressions of mRNA concentrations, and for the BMP2 also CD90, CD105, and there is an increase in the NCAM. There is E-catherine markers increase. There is a decrease in the OCT4 and UTF1, and in ABC G2 conditions also they see an increase. So this results indicates that when this Dasatinib is capable of suppressing the stem cell generations also in a TGF-beta induced conditions. So in the previous video we have understood that Dasatinib can suppress these EMT markers, and in this video we have understood that Dasatinib can also suppress the stem cell generations. Both of them will contribute towards this cell motility or migration of the cells. And overall, they, apart from this checking the few stem cell generation markers, they also check the number of colonies that can be grown from a single cells, and what they are able to find out is that in the case of control, number of colonies is increased compared to the increased in the case where TGF-beta is used compared to the control case. But in the case of where Dasatinib is used, they do not see an increase in the TGF-beta induced cells compared to the control cells, and similar results they have seen for this SB431542, which is the inhibitors of AKT5 pathways. So both of them, or the panel B results indicates that, or it supports the results that we have seen in the panel A, that this Dasatinib can inhibit the stem cell generations in TGF-beta induced cells. And after that they also wanted to check the effect of this TGF-beta on the cell motility. So for this they use the same cell line, it is PANC1 colofit 357, and then they check the relative cell migrations in the presence and absence of Dasatinib plus TGF-beta, and the control conditions is DMSO plus TGF-beta. And what they are able to see is that with Dasatinib they are able to see suppressions not only in the basal migrations but also in the TGF-beta induced migrations. It can be seen in the PANC1 cells and also for the Colo 357 cells. Thereafter they check the dose dependent effect of the Dasatinib, and they are able to find out that at around 0.01 macromolar concentration of Dasatinib, they are able to see 50% reductions in the migrations for both PANC1 cells and also for the Colo 357 cells. And for the control case they use these inhibitors, that is AKT5 pathways inhibitor, that is SB431542-5, and in that case they are able to see a decrease in the migrations both in the control case and also in the case where TGF-beta plus this AKT5 pathways inhibitor is used, that is SB431542-5 for both PANC1 and the Colo 357 cells. That we can say here the migrations is at the bottom level. for the Colo357 and Panc1 cells. So all this indicates that Dasatinib can be used or can be used to inhibit not only the cell motility but also helps in the suppressions of the EMT markers and the stem cell generations that contributes towards these cell migrations. So I hope you understood the effect of Dasatinib in the cell motility and how it regulates the EMT and stem cell generations. If you need to understand more about these experiments or this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.